Okay, welcome to topic 5.6, the final topic concerning genetics. Um, I do want to mention a couple of things. In this one, I pulled this image out here. I want to show you kind of how chromosomes contribute to the overall genetic diversity of not just an organism, but a species. Um, so, the first law, way back when we were talking about Mendel, one of the first things he observed was what's now known as the law of segregation, meaning that the two alleles for each gene separate during gamete formation. So, let's say that we're dealing with we'll do R and Y, just like this one here. R, little r, big Y, little y. What it's saying is that these two alleles will separate. This is law of segregation. Law of segregation. Mendel's other law, the law of independent assortment. The alleles of genes on non-homologous chromosomes assort independently during gamete formation. What that means, and I'll do this one in blue here, is that assuming that R and Y are on different chromosomes, like it's shown here, this is the law of independent assortment. What it means is that these two genes are going to separate independently of each other, right? So we can see that here. We know that eventually, during this meiosis process, you're going to end up with these gametes that are completely unique, and this is due to the fact that these separate genes. And keep in mind, if you'll notice see how these non-homologous chromosomes over here. Can you see how the mother's is on one side, and it's kind of opposite in this case on the right over here? That's the law of independent assortment at work, right? And the fact in meiosis 2, when you get over here and you see that these allele, or actually this anaphase 1 right here, you can see these alleles actually split up at this point, right? Big R splits from little r. So these are these two laws, and there's something that I think is important to know and understand to an extent. And it would help you, especially when answering an FRQ, to be able to say, oh, due to the law of segregation or the law of independent assortment, you understand that the alleles for genes for one gene separate from each other, and also that the alleles of genes on different chromosomes don't. Now, you should be able to tell me an exception to this rule, which we talked about earlier. Go ahead and pause and tell me if you can come up with what the exception to this rule is. You should have said something, if you're back already, should have said something to the effect of genetic linkage. If genes are too close together, they don't really separate from each other, right? They kind of stick together, and they still segregate, but they stick together. Okay. So next thing I want to talk about is something called non-disjunction. Non-disjunction of chromosomes, which can happen. You guys may have heard of situations where this has happened, but up here we have a pair of pair of homologous chromosomes. These are this left side over here. We'll see some non-disjunction in it, but not in this first phase. In this first phase right here, meiosis 1, we have a non-disjunction event. And what that means is that you have these chromosomes not properly separating correctly, right? In other words, these spindle fibers, for some reason or other, end up pushing or pulling these into the wrong side of this cell, right? And then when it eventually splits, you have a cell that is completely lacking one of these chromosomes. This is called a non-disjunction event. And if it occurs in meiosis 1, this is kind of what you end up with over here. A cell with an extra chromosome, and a cell over here that lacks a certain chromosome. Keep in mind this is a very simple system because we're just dealing with a pair, one chromosome, or two chromosomes that are homologous, right? On the left side here, we have a non-disjunction event occurring during meiosis 2. And then you're going to have something similar happen where you have the chromatids being pulled into the same spot. You end up with one that has an extra chromosome and one that is lacking a chromosome right here. So if you'll notice, non-disjunction in meiosis 1 is going to cause no viable gametes over here. No viable gametes. If you have a non-disjunction event in meiosis II, you can still end up with 2 normal gametes over here, if you'll notice. So question is, how does this affect fertilization? Well, you have your sperm. You'll notice it's carrying this little blue chromosome in here. And if you remember, you want them to have total, when they're finished, you want to have 2 total chromosomes in each of these. Of course, keep in mind, this looks like an X up top because it is replicated DNA. Down here, unreplicated, we should end up with just 2 chromosomes. Okay, so you'll notice right here we have 2N. That's right, that's diploid. Over here below that, we have another 2N individual. Then we have 2N plus 1 right here. That means we have an extra chromosome. 2N minus 1, it means we're missing a chromosome. Of course, this is called, you guys may have heard this word before, but this is called a trisomy. Trisomy. That means you have 3 of a chromosome. We'll just put CHR, 3 of a chromosome. You also have monosomy, which is 1 of a chromosome, right? Now, of course, but in most cases, if this occurs, it's not going to be viable. It's not going to divide, right? So in most cases, these will not divide, right? If they are if it's a monosomy or trisomy situation. However, if it is a small enough chromosome with enough, that doesn't code for so much of the genome that it's a massive problem, you can have this occur. And you guys have probably seen something like this before. This is called a karyotype. And you'll notice in this human karyotype, you have 23 chromosomes, 23 pairs of homologous chromosomes, rather. First thing I'd like you to tell me, glancing at this, is it male or female? You should have said male because it's an XY system here. There's also 2 of each chromosome. Why is there 2? 1 from mom and 1 from dad, right? For each of these. Now, I want you guys to look at this second karyotype over here, and I want you to tell me if this is a normal karyotype as well, if there's something wrong with it. Go ahead and pause your video. And let's come back. First of all, I'll ask you, is this a male or female? Of course, you should have said female for this one because it has 2 X chromosomes down here. You also should have said there's 3 of chromosome 21 over here. This is called trisomy 21. This is also responsible. This is commonly known as Down syndrome. Down syndrome, right? You've probably heard of this. Have very distinct features in individuals that have a trisomy 21 situation. There's many other versions of this that can occur. Of course, keep in mind, you have a lot of coding regions and a lot of these other chromosomes that they code for too much. That most of the time, if you get an extra chromosome 2 or extra chromosome 1, that you'll never even notice it because that the fertilization won't really occur. But again, with some of these chromosomes can be duplicated, and you can have a viable fetus. And especially with these X and Y chromosomes, there's a lot of interesting sex-linked disorders that are due to a non-disjunction event. So I advise you to go research some of those things like Turner syndrome or things of that nature, and find out about how they develop, because it's actually pretty interesting. But that's it for chromosomal inheritance. I want you guys to understand what these karyotypes kind of look like, maybe how to look at one and tell whether there's an issue somewhere, as well as understanding what a non-disjunction event is. Please know what a non-disjunction event is. And also know the law of segregation and law of independent assortment. Okay, that is it for genetics. I hope that helped. And let me know if you need some further clarification. All right, thank you for listening.